Most species with separate males and females have two structurally different sex chromosomes, in which one sex is heterozygous. It is much easier for a selfish genetic element to arise on a sex chromosome than on one of the autosomes, because of the lack of recombination between certain regions of the sex chromosomes. The selfish gene will influence the genes that determine the sex of the organism and, as it spreads, distort the sex ratio of the population. The wood lemming, Myopus schisticolor, has a dominant feminizing X chromosome. A feminizing X chromosome drives by reversing the sex of their host. An XY male is converted into a female. These XY females are fertile and produce feminizing X-bearing eggs only, so their offspring consists completely of daughters. This is caused by a double non-disjunction in meiosis, in which both feminizing X chromosomes go to one pole and both Ys go to the other. The YY daughter cells degenerate, and only the daughter cells with feminizing X's proceed through meiosis. The sex reversal in Myopus schisticolor is caused by a deletion and an inversion in the feminizing X chromosome compared to the normal X chromosome. The differences in the two X chromosomes can be seen with a technique called G-banding. G-banding is the treatment of chromosomes in the metaphase stage with trypsin. This enzyme partially digests the protein, and then a chromosome is stained with GEMSA to create a karyotype as seen in the figure. Because the GEMSA stain attaches itself to regions of DNA with high amounts of adenine-thymine bondings, dark bands occur in these regions. AT-rich areas are poor in genes. The karyogram shows that the differences between the feminizing and the normal X chromosome are only present in the short arm of the chromosome, referred to as P. Researchers found a deletion and an inversion on the short arm of the feminizing X chromosome. This was done by different techniques. First, the short arm of the feminizing X chromosome was isolated with a technique called Chromosomal Microdissection. The chromosomes are treated with chemicals, so the chromosomes reach metaphase, in which they are highly visible. Then, under a microscope, the scientist removes the piece of interest of the chromosome with a very fine needle. After the removal, multiple copies of the isolated DNA are produced by a technique called Degenerated Oligonucleotide Primed Polymerase Chain Reaction. This technique uses a primer which binds to multiple places on the short arm of the chromosome. A primer is a small part of DNA that is used as starting point for polymerase chain reaction. So, in this way, the arm is amplified in the form of multiple small pieces of DNA. To all these small pieces of DNA, a fluorescent probe is attached, and all these probe pieces of DNA are hybridized with a normal X and a feminizing X chromosome. Now, the chromosomes are visualized with a technique called fluorescence in situ hybridization. The arrow indicates a gap where no fluorescence is seen. This lack of fluorescence indicates an additional DNA sequence in the normal X chromosome, which suggests a lack of this DNA sequence in the feminizing X chromosome. Later, the same techniques were used to amplify the region P21 to 23 of the normal X chromosome and visualize this region on the feminizing one. No fluorescence was visible, so this part of the chromosome was deleted from the feminizing X. Aside from the deleted XP21 to 23 region, the feminizing X chromosome also holds another characteristic, which is the inversion of the XP24 to 26 region. Researchers found this inversion by creating a probe for the XP22 region from the feminizing X chromosome. Note that this probe is created in the same way as the probe for the short arm of the feminizing X chromosome. Then, the feminizing XP22 probe was hybridized with a normal and with a feminizing X chromosome. After visualization under a fluorescent microscope, the probe creates one fluorescent band on the feminizing X chromosome, indicated by the single arrow. On the normal X chromosome, the probe creates two fluorescent bands, indicated by the two arrows. Two fluorescent bands on the normal X chromosome means the feminizing XP22 probe hybridizes on two places on the chromosome. The only way in which this double hybridization is possible is if one fluorescent region moved to the other on the feminizing X chromosome. This was done by an inversion of the region containing a part of XP24 and XP25 and 26. Since the XP21 to 23 region is deleted in the feminizing X chromosome, it is interesting to find out which genes are on this region. In this way, One or several genes may be found which are involved in the sex reversal of the wood lemming. The approach to find these genes is to compare the genes of the wood lemming in the XP21 to 23 region to genes in mice, since there is much more known from mouse genes than from genes of the wood lemming. First, researchers created a cDNA library from genes expressed in mouse fetal gonad cells, since genes on the X chromosome are normally active in these cells. In this technique, messenger RNA from mouse fetal gonad cells is isolated and transformed into cDNA by reverse transcriptase. All these pieces of cDNA correspond to all the active genes in the mouse cells. The pieces of cDNA are cloned into vectors and transformed into E. coli cells. The cells are grown on agar plates, and each colony corresponds to one piece of cDNA, so to a certain mouse gene. So now, all the active genes in mouse fetal gonad cells are present on the agar plate in the form of bacterial colonies. A disc of absorbent paper is added to the agar plate. The colonies stick to this paper disc, so after peeling off the disc, a replica of the agar plate is produced. Note that every colony on this replica represents one mouse fetal gonad gene. The bacteria on the paper disc are now lysed, so only chromosomal DNA and the plasmid containing the cDNA molecule stick to the paper disc on the place of the original bacterial colony. The paper disc is now incubated with radioactive labeled small pieces of XP21 to 23 wood lemming DNA. When a sequence of the wood lemming probe is identical to the sequence of a certain mouse gene on the agar plate, they will bind. The homologous genes and their place on the agar plate will be visible after exposure to a photographic film due to the radioactive label of the probes. Now, the cDNA can be isolated and sequenced. The gene can be determined by comparing the sequence to a mouse gene database. This technique yielded 14 potential homologs. After sequencing of these DNA and comparing it with the mouse genome database, 3 of these positives turned out to have no homologs in the mouse. 10 others had homology to gamma satellite DNA, which is a piece of DNA consisting of repeated DNA. The final candidate is the most interesting, since it had homology to the mouse CCTH gene, a gene that is mainly expressed in testes.